לא ישנתי 48 שעות.
לישון ולחלשים.
חמור. אם אני מדבר שטויות, ואו נרדם בעמידה, תעירו אותי ואו תתקנו אותי. אתם יודעים שאני מדבר שטויות. Okay, English, we move to English. Good morning. Hello, I just told everyone that I am in physically bad shape today because I have not slept for many hours. I just landed 1 hour ago from the US. So if I will sleep while standing, please wake me up and or say wrong thing. Today we're going to talk about, we're starting a new sense, the chemical sense, the classical chemical sense, what is known as olfaction. We'll see how we go along. I doubt we will get to taste, I don't think so. But we will stick to the olfactory system first, and we'll see how we develop. Okay? And olfaction is evolutionary ancient sense. You can find it in worms, flies, and many animals along the evolutionary hierarchy. It's a central sense in general. And if you look at the whole animal kingdom, it did suffer a little bit because in humans it's degraded somewhat in non-human primates and humans. But nevertheless, it's a very robust and important sense from fish, snakes, flies, mammals, etc. Okay, in the sea and in the air. For humans, it's a sense that is evolving around the food industry and the perfume industry, which there's a lot of money there. But frankly, there's very little interest in for these companies in real scientific, real academia. There is some interactions between academia and the industry in this sense, but it's not too fruitful in my eyes. Okay, but nevertheless, there's a lot of money around the olfactory system in economical terms. But for other animals, like everyone who has a dog surely knows that sniffing and smelling is a very central sense. And also for the animals that we study, the model organisms in neuroscience, like worms, flies, and mice, this is very central sense. Okay, there are some people, like my colleague Noam Sobel from the Weizmann Institute, that argue strongly that this is a very central sense in humans as well, so I cannot say it is not a central sense in humans. This is an image when he was in Berkeley. He did an experiment. On the right-hand side, this is a well-known image of a hound dog, which is released to find the smell of the chicken, of a dead chicken, that was dragged along this field. And you can see the pathway of the smelling hound dog. A few years ago, in my lab, we work on olfaction. So we work on olfaction and audition. So this is my bread and butter, if you will. A few years ago, we went, me and my graduate student and I went to the police office here, Magab. They're training dogs to find drugs and bombs. It's unbelievable to see these dogs, how they can find very tiny amounts of chemicals. Anyway, so this is a famous image of it, a hound dog looking for chicken. And what Noam did in his early stages of the career, this is a student in Berkeley. He paid them money to trap chocolate on the grass of the campus while shutting down the other senses and trying to prove or to do some analogy that human olfaction is powerful. And whenever you will hear him, and you will if you will going to be part of the Israeli Academy, this is something which he tries to promote very strongly.
There was some article, I think, about the fact that the shaking hands between people is
Noam has many great papers. So a few years ago, the nicest one I like is when Yair Lapid was the anchor of Channel 2. So the last item on the news was, and he's so beautiful and dramatic, Yair Lapid. Why are people crying in the White House? That was the heading of the item. And what they did there is they collected tears from girls or women that they showed them the movie The Champ, Pro Shel Aluf, I don't know if you know it. It's a very heart-wrenching movie. The little boy that his father is a boxer and he dies and everybody cries. So they brought students to the lab, and especially women students, and then they started to cry, and they took Eppendorf and they collected tears, and they did an fMRI study of the odors that are in tears and how they affect all kinds of regions in the brain in males. We'll talk about that also. I think we'll reach that. There's great stories about tears, odors in tears. Anyway, so you'll find that. Yeah, the latest one was shaking hands that people have to smell. But let's start early, okay? This is one of the most dramatic stories of olfactions is olfactory imprinting in salmon fish. And if you know, one of the known behaviors of salmon is they go back to the river where they were born to spawn there, and then they die, they spawn, and life begins again. And one of the nice papers that showed this is this field study from 1976, where they grew salmon in 3 types of ponds, okay? Artificial ponds outside the real rivers. One was a control, just a regular pond with fresh water, and 2 ponds with 2 different odors. One noted here as N, morpholine, and one noted here as PA. And then they took the salmon and they let them loose into Lake Michigan. And later on, they put these scents, these 2 scents, the PA and the M, in 2 specific rivers, tiny rivers. They put the scent in the river and counted the amount of salmon from their origin, where they came from. Okay? And as you can see in this table, I normally don't put tables in slides. It's not a very aesthetic thing, but nevertheless, this is a very clear example of the number of fish that they caught from the original ponds in the different rivers. So then the field study was that they collected the salmon at the end of their life at all these rivers, and these 19 different rivers, and they collected the number of fish. You can see that control fish were distributed in almost a random manner, and PA, which was put in the 2 rivers breakwater creeks, the vast majority, 118 fish out of, I don't know, about 150 were collected there, and the M was put in the little Manlitauet River, and 207 fish were collected there from the fish that they released to the river. This is a very strong and well-known example of an imprinting in fish, and indeed the odor that they experience during young life has a strong impact when they return to spawn. There are also examples in mammals, especially the smell of the mother, the smell of the nest, the smell of home, etc., which has a strong impact later in life, and there are many examples of that in the literature.
What in the odor drives them to the place?
What do you mean, what in the odor? What is the mechanism, the neuronal mechanism in the brain, you mean? So this is the cue that they learn. All I'm saying is that this is a course about sensory systems, okay? Today we're starting on olfaction. And I'm telling you that olfaction, many animals in the animal kingdom has a very strong impact. One of the strongest stories is the imprinting story in fish. Okay, there are also other imprinting stories. In vision, for example, you may know the example of the chicks, and they see the image and they go like, very cute ducklings, they go after the mother. This is a visual imprinting. This is an example of olfactory imprinting. And the smell of home, when they're very young, is the cue that they remember throughout their life. Yeah. But we don't understand the neural mechanism in the brain of sun.
What's the word imprinting in Hebrew?
Achtama. In Hebrew, achtama. Okay. So from fish we jump to humans, and this is just an anecdote. I don't want to dwell on this too much. About how many odors can humans smell? Can we smell? Okay. Again, I apologize for my physiological shape. And the estimations were that humans can smell about 10,000 odors, roughly. That was the ballpark, the number of odors that we can smell. But the truth is that we know very little about this. And in Science 2 years ago, a year and a half ago, there was a paper entitled "Humans Can Discriminate More Than 1 Trillion Olfactory Stimuli." It's a group from Rockefeller, Andreas Keller and Leslie Goswald was here last year or 2 years ago in the rainy days. And what they did is they did an experiment where they let people smell two different mixtures of odorants, which was a combination of 10 monomolecular odorants. These can be completely separate 10 monomolecular odors, thus there's no overlap between odor A and odor B, and they asked the people whether they can smell them. And they started to do mixtures of these odorants, where they, for example, have 3 of the 10 components in the two mixtures which are exactly the same. Okay? That they would call this 30% overlap. They can have 6 out of the 10 overlap, or 9 out of the 10 components in each other overlap, and that's a 90% overlap with only one component different in the mixture. This for sure will be much harder to discriminate, right? Because these odors are 90% overlap, and it will be hard to discriminate this. And they did it with mixtures of 10, 20, and 30, and asked human subjects to discriminate. It was a 3 alternative choice force task. I won't elaborate on that too much. And again, these are the mixtures. Chance in this case is 33% because it was a 3 alternative force choice. And you can see this is the mean. The red is I think above mean. Yeah, those who can discriminate statistically, and the black ones is that they cannot. And this is the hard mixture, and the easier it gets, the higher the percentage of success. This is a basic psychophysical experiment, and different curves for the 10, 20, and 30 components. Of course, 30 components is even better than the 10 components if they are separated, and when they're 90% alike, or here it's more than 96% alike, nobody can actually discriminate that. And from this, they calculated all kinds of estimations on how many odors can people discriminate, can humans discriminate. And this made it to Science. Science is a very, as you may know already, very high-profile paper that makes the news, is that they claim now that smells, people can smell about 1 trillion odors. And the estimations for colors was 10 to the 7th, and to tones and sounds was 10 to the 5th. So olfaction is a great sense. Soon after, no, we're not going into this paper. Okay? This is just a teaser, because soon after, in bioarchives, a few months later, came Marcus Meister from Caltech, and I'll just read the abstract of the paper. A recent paper in Science magazine answered this question in the affirmative. Okay, how many smells can humans discriminate? The authors reached that conclusion after performing only 260 comparisons of 2 smells, of which half could be discriminated. Furthermore, the paper claims that human ability to discriminate smells vastly exceeds our abilities to discriminate colors and musical tones. Here I show that all these claims are wrong by astronomical factors. A reanalysis of the author's experiment shows that they are consistent with human discriminating just 10 odors. The paper's extravagant claims are based on errors in mathematical logic. Okay, so those of you who do want to, so this was a very strong claim. Apparently he's right. Mathematically he's right. They made all kinds of mistakes there, and they admit, although it's still a highly debatable topic in the field. Yeah.
Where was it published?
So initially he published it in Archives. Archives is
It's a forum.
exactly. So many people, they just want to, they don't want to start with peer review. So initially it was published in Archive, made a lot of buzz in Twitter and Facebook, etc. And last year they published 2 back-to-back papers, Meister and another group, showing that they are wrong in Eli. And that was peer-reviewed, etc. But the field argues that there were some mistakes in the other paper. Of course, the authors of that paper claim that to some extent they are right. But one thing for sure is wrong is this comparison between the sound, the smells, and the other senses, which is, with all due respect to olfaction, in humans, it's wrong. Okay, so we can.
So in the current opinion, where do you put the actual number? Like, is it close?
So we don't know.
100,000,
We don't know. We have no idea. Okay, and the problem is that this leads me to the problem. Okay, but before we go to the problem, I just again want to tell you. So what? We don't know how many odors can humans smell, nor any other animal in that sense. And also, it's a very young, in terms of research, it's a very young field of research. So we know far more about visual system and auditory system, etc., than we know about olfaction. So this is my non-academic survey. So you can see 100 pages in the textbook of Kandel versus 8 pages on olfaction. This is a review from 1965, just talking about olfaction, saying, let us suppose in the very limit that we have utter chaos. 1965, we don't know anything about that. 2006, that's 10 years ago, were much better. The study of olfactory processing may no longer be in its infancy, but it has yet matured beyond stormy adolescence. Just to tell you that we don't really know a lot about olfaction, and we do know only a little bit. What we know is in the very early stations of the olfactory systems, and this is what we're going to talk about. You had a question here? No. So we're starting from the odors. That's what we want to understand, like any other sense, from the chemical stimulus to the perception, and we are really here without knowing almost anything about the odors themselves. Okay? And why is that? And why don't we know how many smells can humans smell? Because we do not understand odor space. An odor, if you take a natural odor, pick an odor, normally it will be composed of 10, 50, 100, 200 different molecules. Okay? So it's a combination of molecules which can have very different structure. Some of you who studied chemistry may be more understand better in organic chemistry than me, but these are very different structures. Okay, so you can see very, very different molecules that are composing any odor, almost in nature. So the first efforts to try to define odors is to put them in some kind of quantitative framework, so we can handle it. We can start to understand it. I think I had a slide in the introduction of the course, and this started by, as many, many things in neuroscience, by psychologists who initially defined all the odors in the world into 7 categories. That was good for 100 years. Then came another psychologist and decided there are 9 categories of odors that should be. And then there's the מצרה של הנס הנינג, מצרתי של מצרה. מצרה. Prism of Hans Henning. We said no, it's not different categories, it's actually a prism, and any order should be somewhere on this three-dimensional space. So it's a little bit spicy and 20% burnt and 10% foul, etc. Okay, so we can categorize that. Today there are much more advanced ways of looking at odors and molecules. We'll talk a little bit about that as well. But the bottom line is that if you compare vision and you compare audition, these are stimuli with a very clear space of the stimulus. Okay, so we can characterize sounds, we will talk about that when we talk about the auditory system, by a very clear physical measure. We can look at light similarly, okay, in terms of wavelength, intensity of light, etc. Others, it's much more complex. So people are, for example, have been using different chemical groups, okay, whether these are aromatic rings and the number of carbon atoms in the molecules, etc., etc. But those are not, they don't always fall correctly, okay? So a molecule with 5 carbons versus 6 carbons sometimes can smell perceptually different. So the question is whether, and for us, if we see 5 kilohertz or 6 kilohertz is more similar to 5 kilohertz, and we can actually scale it perceptually quite easily as opposed to, yeah.
Why not work on this from the viewpoint of receptors? I mean, instead of trying to categorize all possible forms of molecules in the world, see which receptors the human nose has and like what function.
That's where, you know, it's a good question, but you are leading me a little bit ahead. The story evolves. Yeah.
Just in addition to what you said.
But it still did not solve anything else. Okay, let's say you have 2 receptors in the nose. Or
if you start
5
with the
receptors per
structure,
odor.
you know what's the ligand site, and then you know which kind of chemical can activate it.
Okay, but then you have to sample all space. And space is infinite. Infinite. In auditory, I can, you know, go from X kilohertz to Y kilohertz within the range
But
of the
also,
animal.
it's a complement, it completes this picture because
Yeah,
I mean,
okay.
we'll at least know what we're looking for. We know the binding site in a way. It's in like the structure becomes less complicated even or more specific.
Not exactly, because as you will see, odorants can detect substructures on a molecule. So it's not a full molecule.
Yeah, but you still have a substructure.
Yes, this is not, I'm not sure how can we define outer space completely like that, and it may be different for different animals completely. And I'm not complete, I think you can have limits on that, on the space, but again, defining the space in a clear,
Yeah, it has to be done this way.
I don't know if it has to be, but we still don't know how to handle the space today. Yeah.
Just in addition to what you said, perhaps you can use not the input, not to analyze the input and how it's connected to the components that gives the neural reaction, but you can perhaps analyze the space by just getting the output of the sensors. Let's say you have some.
What is the output? The spikes of the sensors or the perception?
Exactly. The spikes.
Okay.
The perception is what the psychologist would say, but let's say the
So
spikes.
this is again another work of Noam Sobel, and he claims life is very simple. There's one axis of perception of odors, and that is the valence of the odors. Okay, so you can scale odors from very good to very bad odors, and that's a clear scale because the primary dimension of olfactory perception, in principal component 1 space, whatever, is reflected in odor and pleasantness. So you can take 100 people, ask them to smell an odor, and they will tell you if it's pleasant or not on a scale from 1 to 100, and he says that this is a clear metric and scale that you can scale odorants. What he also claims is that when he tested neuronal physiology of specific smells, they also categorize nicely in principal component space as pleasant. And when he looks at the actual component, and you can take today in chemistry, you can take a molecule, put it into an analysis, and you can get 3,000 descriptors, chemical descriptors of this molecule. Okay, things that make no sense to me at all. So boiling point, the melting point, the aromatic ring structure, the number of hydrogen connecting to whatever, beyond me. This principal component specifically of pleasantness is the eigenvector of the molecule at 200 Kelvin when something happens, I don't know, something bizarre completely which doesn't make sense, but still perceptually and physiologically he claims it's very clear.
And is there no correlation between this axis of Kelvin to physiological response of
To the chemical structure of the odors and physiological response. Now the correlation is low, it's about 0.4, something like that. But for psychology, it's good. Yes, psychologists, 0.4 is a very high correlation, so that's good. Yeah.
What are the other principal components? I mean, that's very nice, but if he has...
It will tell you nothing. So the first, if you take the first different principal components, the first 3, 4, 5, which explains 90% of the...
Maybe into, I don't know, how much it smells flowery versus like some more accents.
No, so he cannot find any other perceptual correlates which they tested, and this is very strong part of their work. Okay, so they're still, again, my point here is to tell you that we are in the early days of olfaction. We're still struggling in defining what is an odor, what is odor space, perceptually, etc., etc. Okay, so this is where there are many open questions from the side of the molecule and from the side of perception that we're still in very early days. Okay, but now we're going to start to listen about what we do know biologically very clearly. Okay, so we don't really understand odor space, but whatever chemicals come in, we do know it's volatile molecule, up to about 300 molecular weight, which we can smell. Anything beyond that, we cannot.
What's volatile?
Volatile, nadif.
Oh.
Okay. There are different molecules which are not volatile, which not we, but rodents, for example, can smell. These are called pheromones. Or again, I'm not sure we'll have time to go into that. So when you smell, stream of air goes into your nostrils and hits the epithelial surface. This is called the olfactory epithelium. It's actually a very large region in the nose of rats, for example. I'll show you. I think I have we can jump ahead to okay, so just so you get a feeling. I think I showed this also. Anyway, this is part of the head of a rat. This is the front tooth, this is the nostril, this is the upper palate of the mouth. Okay, sorry for the aggressive image. And this rat is spliced in half. What you can see is the brain. Here, that will be the olfactory bulb, that will be the cortex, the cerebellum, and the brainstem, and the spinal cord. Okay? So the whole brain is something like this. This is the sensory epithelium. It's about at the size of about 80% of the whole brain in terms of width and height. Okay, so it's a very big epithelial structure. Okay, it's not as big as the skin, for example, that surrounds the animal, but nevertheless not like the ears or the eyes. So it's a surface, epithelial surface. It has turbinates to increase the surface area of the epithelial surface. And when you dissect one of these out of a mouse, you see that it looks like a jelly, brown jelly.
How much size does it take inside the mouth? Say that again? What does?
Yeah. So this is the oral cavity of the rat.
So where does the head end? Like it's
again, the head ends here. 25 from the size of the brain.
Oh,
So if
okay.
this is the brain,
This
this is the epithelial cell,
Okay.
50%, whatever. It's a significant portion, okay, in terms of size. So we start there. What is there in the epithelium? In the epithelium, there are different types of cells. Of course, this is the first interface with the chemical world outside. And you can find different types of cells. One cells are big fat cells. They're called support cells. They provide the nourishment that these cells need to have. There's a thin layer of mucus around these cells. And in between the supporting cells are the receptor cells, or the olfactory sensory neurons. So you would see this interchangeably, the receptor neurons and the olfactory sensory neurons. These have a cell body, and they have these dendritic structures, which are cilia-like, which are beating. And this is the odors that are coming in. They hit this mucus in the epithelium, and the first interaction is with the cilia of the olfactory receptor system. This is a very unique receptor cell in the nervous system, in sensory systems, because it sends its axon directly into the central nervous system through the bone. All other receptor cells only have local transmissions. This cell is actually a cell that sends an axon into the CNS, and the cell body and dendrite sits outside the open world. Most of the receptors sit outside of the receptor world, but are very local, like in the retina, the cochlea, skin, etc. Okay, the receptors. Here, the receptor cells are very unique in the fact that they send axons into the CNS.
Is this for all mammals?
What?
Is this true for all
Yes.
mammals? And well, like humans as well as
Yeah,
mammals?
absolutely.
So there not be a lot of modulation on the input because there's
We will see there is modulation on the axons.
on the axon.
Yeah, but generally speaking, there's not huge modulation on receptor cells of the retina or the skin. Or those, their role is to detect the stimulus outside.
But the information flows from the periphery to the brainstem.
Here,
and we have
here
a
too.
lot
There's modulation along the way.
along the way. Here it's shorter.
Yeah.
And there's not a
This is not the only difference. Yes. Okay. We'll see that olfactory circuits go without relays, without thalamic relays, directly to amygdala and brainstem centers, which has to do with the emotions and things like that. Okay. Other systems go through other relay stations, as you have seen, as we will go on. Yeah. More questions? Okay, and another interesting thing here are these cells that are called basal cells, and those are in fact cells that will become neurons when they grow up, along the life of the animal. So basically, this is a tissue that is renewing itself with cells that can become neurons. So those are a source of neurons to regenerate, if you will.
The humans?
Yeah. Okay. This is an EM image of the sensory epithelium. There are also these some proteins called olfactory binding proteins that are thought to shuttle the volatile molecules through the mucus into the receptors. This is an EM image of these of the epithelium. So these huge blocks are the supporting cells. These are the olfactory cells. You can see one that was detached here. It looks like these toothbrushes, you know, the automatic ones that turn around. These don't turn around, of course. But if you look at the surface of the epithelial surface, it's this huge, all these toothbrushes are looking at you. And in fact, it's like a jelly-like because they're beating. So if you look at it on a live tissue under the microscope, it shakes at you. Okay. So that's the epithelium. And the epithelium has millions of cells. Cilia are beating, and there is continuous new supply of new neurons from these basal cells. It's also unknown how long is the lifespan of these cells. The estimates are about 2 months. Okay? And if we'll have time to talk about neurogenesis, we'll get to that.
Isn't that easily checked? Like if you just mark one and then
So some cells will die very fast, some cells will live forever,
oh.
and the average lifespan is about 50 days, but there are very few papers on this topic, and they disagree. I'm one of them. I did some work on that earlier.
Are there factors that can help you predict what's going to happen to the cells?
Not in the first 2 weeks, no, it's very hard. So it's like, I don't know if you learned developmental neuroscience, a little bit. Okay, so there are competitive interactions early on, and depends on trophic factors and activity levels and things like that. But later, after you're integrated into a neural circuit, I don't think anybody knows can predict what will happen, because different things happen to the cells all the time. So we can't predict a priori what will happen. Like I said, we can detect molecules up to 300 molecular weight. They're normally lipophilic. Okay, so when we start to study olfaction, first thing we do as a neurophysiologist, we stick an electrode into the receptors. Right? When you stick an electrode into the receptor cells and you give an odor puff, you get a nice depolarization. This is very different from the visual system, where you get hyperpolarization, but nevertheless you get depolarization and you also get spikes. Many receptors you don't get spikes. Here you get spikes because these have to travel. You remember this, these cells have an axon, so you get the depolarization and spikes given the stimulus here. And the first trials to understand what's happening in the olfactory system was from the side of the receptors. Your name again? Gal. What Gal asked and said, let's start from the beginning, from the receptors, to try to understand even odor space. So surely if you want to understand the nervous system, you want to start at the beginning of the olfactory receptor cells. And the first goal was to find the odorant receptors. And that was a big challenge, not an easy one. But the first experiments were, you can patch the cell, you can put a patch electrode on one of those dendrites of these isolated olfactory receptors, and you get these depolarizations. When you put an odor, I told you before, if you put your electrode in the soma, you'll get depolarization and spikes as well. At that time, early 80s, mid 80s, late 80s, it was the big era of the cyclic AMP signaling in cells in general, and it was clear that these cells are also working through cyclic AMP signaling pathways in the cells, and also that probably cyclic nuclear-gated channels are involved in the depolarization of the cells. Okay, so that was the thought, and it resembled quite a bit the receptors in the retina, okay, in terms of the sensory transduction of what's happening in the cells. So the search was focused towards this region. Okay, so this is one of the experiments. You can increase cyclic AMP by putting this inhibitor of some enzyme in the cell, of phosphodiesterase, and you get a mimic of an odor response. Okay, so you can get that. And the big breakthrough came from Linda Bach, who was a postdoctoral fellow in the laboratory of Richard Axel. Linda Bach actually came to Richard Axel to do a postdoc. It was sort of a second postdoc in early 1980s, 1981 I think, or 82. And after 2-3 years of hard work, she had a paper in Cell, and she was ready to go on to find a job and continue her life, some work on CLA. But she decided she had a crave to find the odorant receptors. And she started the project, which failed. And it took her 8 years to find the odorant receptors. Eventually, she got the Nobel Prize for this discovery. They both got the Nobel Prize for this discovery. For her postdoctoral work, she then went to be, she got a position at the Harvard University, and now she moved 10 years ago or so to Seattle, to the Fred Hutchinson Cancer Research Center. Richard Axel is in Columbia, and he's perhaps one of the leading figures in the olfactory world in neuroscience, and we'll talk a lot about the work of his work today. But mainly the drive for the discovery of the odorant receptors was in the hands of Linda Buck, and we'll see why she got the Nobel Prize for that. So Linda's Buck search, okay, so he had her paper on C. elegans on something, some immuno, she found not extremely interesting, and she was hunting after the ORs. Again, this is the time of cyclic AMP, and she was looking for olfactory receptors, which would be some proteins that would be expressed in the sensory epithelium, and the first years of her search failed. She could not find anything, but again, she was in this era of high discovery in the molecular world. So in 1985, a new technique just became available called PCR. You guys are now experts in PCR after the methods course. And it's the area of the G protein coupled receptors. Okay, those of you who learned physiology, the receptors on the heart, blood vessels, etc., were discovered then. Big thing, the G protein coupled receptors, they are identified more and more every time a Science Nature paper discovering a new G protein coupled receptors. And also in 1988, the G-OLF is discovered. This is an olfactory G protein, only in the olfactory system. So maybe our receptors is a G protein coupled receptor.
How is this protein discovered?
So this protein is identified through biochemical analysis of the different tissues, and they find that it's specific to the olfactory system. We're not going to go into the biochemical methods. I probably don't understand them in depth as well. Okay, but this is the era, and she started a second search using the primers of GPCRs that were known at that time. And she failed again to find any interesting G protein coupled receptors in the olfactory epithelium. So what she did is she designed her own primers based on the latest GPCRs that were discovered. Okay? So she took all the GPCRs known and she designed a set of 11 primers that were similar along all the GPCRs known at that time. Thinking, her hypothesis would be that the olfactory receptor would be a GPCR. Okay? And she took all different pairs of combinations from these 11 primers and ran them on a gel and got 64 bands. Out of the 64 bands, 22 were further analyzed. And what the hypothesis was here is that if she could try fishing these proteins, if she would fish specific GPCRs from the nasal epithelium, and she thought it would be a few proteins, that would be was the initial hypothesis, because different cells would respond to different odorants, and they will resemble the GPCRs based on the protein analysis, it will be a hit. And in fact, out of all the 22 gels here that you can see, number 13 was the hit. And this gave her eventually the Nobel Prize.
How did she
Band on
eliminate
the gel.
the others?
What?
How did she eliminate the others?
So she did not eliminate the others. She took all the others and she put restriction enzymes in them. And the idea was that if any one of these bands is a family of proteins, the sum of the restriction and enzymatic reaction would give you as if there are many proteins of this type and not just the sum of this protein in this type. And out of all these, only number 13 stood the criteria. So when she broke this to its different component, it was not the sum of one product, but many products in the gel. And this means that there are many proteins of a similar kind in this gel.
Like the restriction cut the different proteins in different ways?
Yes. Okay. Normally, if you take one protein and you cut it, you will get, bits and pieces of it, and you sum it up to one protein. Here, she used the enzymes, of course, in an intelligent way, and then she got that it summed up, and surely enough, this band number 13, she cloned this band, and she found 5 different proteins from the GPCR family. Okay. The GPCR family of this 7 transmembrane domain that has, that passes the membrane 7 times, and this was restricted to the olfactory tissue by these northern blots and not the rest of the brain, heart, kidney, liver, etc., etc. Okay? So this was the discovery, and in their seminal paper from 1991, they cloned 18 members of this family, and based on all kinds of estimations of how poor their primer sequences is, they estimated there's a minimum of 100 genes of olfactory receptors. So this is a huge multi-gene family of receptors.
Wait, what? Can you repeat why does this mean that it's a different gene and not doesn't alternative splicing or something like that?
No, it's a complete, because you did the cloning. You take the cloning, you take this protein, you can do the cloning of this DNA, you can find that it's a completely different gene. It's not in an alternative splice gene, it's 5 different proteins that she fished with her techniques. And these 5 different proteins are all in the olfactory epithelium and all members of a GPCR family. The
So
cell
how can you say
transmembrane
that
domain.
there are at least 100?
So this is based on all kinds. They cloned eventually 18. This first band was 5, but then they did it again, and generally were successful several times, and they got 18 different members of the family. But every time you take the tissue, you fish different receptors. And they can do calculations on roughly how many receptors they think there are in the epithelium. And the discovery, the big news was that it is 100 genes. Okay? If you look in the visual system, we have 3 receptors. 1 for light intensity and 3 for colors. Here we have 100. And in fact, this was a minimum bound. And very soon after their paper, they found that there is 1,000 genes of odorant receptors in the genome. So you should now wake up from your Passover vacation and say, what? 1,000 genes in the, how many genes we have in our genome?
20,000.
20,000 genes. Out of the 20,000 genes, 1,000 is dedicated to olfactory receptors. This is the largest number of gene family in the genome that we have. The only one that competes with this is the immunoglobulin, the immune system. The immune system, it makes sense. This is the army of the body. It has to fight for the nose, 1,000 receptors.
It makes more sense when you look at the evolutionary perspective. I mean, for mice, it's much more prominent to survive than it is for humans, and just stuck around.
I don't understand this rationale. For me, it's still surprising why we use so much molecular machinery to detect this. Okay, so we can do beautiful things with vision, conceptually, whatever. No, mice can also with the sounds, whatever. Okay, take us. We don't have 1,000. We have 400 receptors. But still, we have 3 in the visual system, but we have 400 for the olfactory system. 400. Now, maybe this has to do with dealing with the vast odor space. I don't know. But the fact of the matter is that a huge part of our genome, of our DNA, is devoted to olfactory receptors. And this is why they got the Nobel Prize. Yeah.
Only for receptors or whole olfactory system?
No, only the proteins that sit on the sensory neurons in the nose.
Maybe they have other uses in the body?
Okay, we will talk a little bit of other uses. Generally, no, not much. You can't find them. I think there's a few examples out of the 1,000 that you can find in the gut or something like that, but generally speaking, you cannot find them anywhere.
But why doesn't it make sense evolutionarily? Like, if the first sense that really developed was this, and the molecular machinery to create alternative splicing and stuff like that wasn't so effective yet, so like ancient ancestors of mammals developed a large family of odor receptors, and it stuck there because it didn't matter, and it just like the gene just grew.
Maybe. That's an hypothesis. I don't know. Maybe I don't know. It's an hypothesis, but for me it doesn't strike. I mean, our body has to do many things. Okay? Smell is tiny bit of that. For a worm, maybe not. A worm has, I don't know, 50 genes of olfactory receptors, or 60, I think. But still many thousands of other genes. So sorry. Anyway, this is the paper. I see you're moving uncomfortably. We're going to go to a break in a second. This is the paper, 1991 paper of Bach and Axel. It's the most cited paper of both scientists, over on, I think now this is 4,000 times. A huge, huge impact in the field. In fact, in our LSEC retreats, we have debates, artificial debates. I don't know if you were in one of them. So every time there are two faculty that fight with each other fiercely. And I had a fight with Chagai Bergman. Chagai had to defend the position that the only way to understand the brain is with electrophysiology. And I had to defend the claim that you don't really need electrophysiology to understand the brain. And my argument was that people have been sticking for 20 years electrodes into the olfactory system and understood nothing until this molecular survey that opened our eyes onto how the system works, and we'll see how it really changed the field of olfaction, this discovery. Okay, and of course it got them an award because this is really a huge gene family in the genome that opened the pathway to study olfaction. Let's see where we are. Yeah, just a few words on these receptors, and then we'll go to a break. So this is the first, 1984 version of what we knew. There's depolarization. Now we know there's a whole intracellular cascade. An odor molecule will bind to an odor receptor, 1 of 1,000. This will activate the specific G protein of the olfactory system through the cyclic AMP classical pathway and open cation channels for depolarization. In fact, we now know also that the calcium and sodium that goes into the cell will open a chloride channel, which will further depolarize the cell. Yeah.
Did I get any one molecule or combination of molecules?
Excuse me?
Did I get with regards to the receptor?
Uh-huh.
Was it one molecule?
No, it's actually not even a molecule, it's a motif on a molecule. Okay? And when you knock out these receptors, these animals become anosmic completely. So if you make a knockout mouse without this channel, the whole system shuts down and the mouse becomes anosmic. Okay? And let's take a break and then we'll continue to talk about these receptors more. Then go on to inside the nervous system.
מחשבה. כן, אני יודע שזה לא יהיה התחום שלי, כי אני פחות של אלקטרודות, אבל נניח, אם אני חושב שעשו את זה, לקחו כאילו output של הרספטורים, וניסו לעשות להם איזשהו קלאסטרינג כדי לקחת, להתייחס להם בתור איזשהו וקטור, ולעשות לרחבים שונים, לעשות איזשהו קלאסטרינג לגבי הפעולות, ולבדוק כמה קלאסטרים שונים יש לרחמים, כמו 5 דברים כזה של...
לא הבנתי.
פסיכולוגיה.
לא הבנתי.
כאילו להשתמש באינפורמציה, באוטפוט של
של מה? מה
האוטפוט של הרצפטור. של הרצפטור באפיטליום.
של הספייקים של הרצפטורים.
כן. לנסות להכניס אותם לקלאסטרים שונים, את שונים של...
הבעיה שלא כל רצפטור יגיד לכל הריחות. יש לך 1,000 רצפטורים שונים. אתה עכשיו צריך לבנות את התגובתיות של כל התאים וכל הריחות. כל הריחות יש לנו בעיה להתמודד עם המושג הזה בכלל, כל הריחות. אוקיי, כשאתה לוקח פוטורספטור ואתה פוגע בו עם פוטון, אתה מקבל איזושהי פעילות. או שאתה מדבר על הרסל בקוקליה, אז אתה יודע, כשיש עוצמת תלות מסוימת, יש דיפלקשן של השערה, תלוי איפה יושבת, הרקנה תפוי לזה, אתה יכול start to make sense. פה תמונה קצת יותר מורכבת.
כמה סדרי גודל.
כן. זה סכום שהוא קצת בחתולים בגלל המולקולות העולות האלה. עכשיו, האמת היא שב-20 שנה האחרונות היה הרבה הייתה הרבה התפתחות אצל הכימאים. והם יודעים להתמודד עם מולקולות בצורה הרבה יותר רצינית ממה שידעו פעם. אבל אין הרבה אינטראקציה בין נוירוסיינטיסט וכימאים. בניגוד למדעי המחשב ופיזיקאים שבאמת קידמו את התחום של ויז'ן.
קצת רקע
ומה?
בכימיה.
כימיה אורגנית.
כן.
גם לי. אני פעם אחרונה שעשיתי כימיה אורגנית היה בשנה א'.
אז אני...
20 שנה.
אני לא...
אתה לא עשית בכלל?
כן.
אתה לא צריך לדעת, מה אתה צריך לדעת?
אני לא מבין למה האלקטרודות לא עובדות?
מה זאת אומרת האלקטרודות?
אמרת שהם ניסו 20 שנה, כמו שאמרת, לחבר אלקטרודות...
לא, אמרתי שרשמו מהמערכת של הריח. כן, זה כן עובד, אתה מקבל סיגנלים, אבל לא הבינו את המערכת לעובד.
אז לא הבינו מה עושה את הטריגר.
בדיוק, לא הבינו את המנגנון עצמו, איך הרשת עובדת וכו', ובעצם התגלית הזו, המולקולרית, שינתה את התחום.
ממש יש לך 1,000
יש לך מיליונים, 2 מיליון תאים. אוקיי? 2 מיליון התאים האלה, גם ברטינה יש לך 2 מיליון תאים, אבל כולם מבטאים את הרודופסין הזאת, אוקיי? את הרצפטורים שרגישים לאור, או לאורך גל מסוים. פה אין לך 3 רובע, יש לך 1,000. אנחנו נראה אם הם מתבטאים בכל התאים.
איך זה מתבטא בשונות? מה השונות בהם?
חלבון אחר, לגמרי.
רספטור זה חלבון?
רספטור זה חלבון שיושב על הממברנה ומזהה את הסיגנל, הוא ממיר את הסיגנל הפיזי לסיגנל ביולוגי.
זה חלבון.
כן. הוא חלבון שיש לו אינטראקציה עם העולם.
אז יש 1,000 חלבונים שונים?
חלבונים שונים. לך בגוף, בכל תא בגוף שלך, יש לך DNA, ה-DNA זה הספר של כל החלבונים, ושם יש 20 מהם. כל תא מבטא משהו אחר, לא כל תא בגוף שלך יבטא את כל ה-20. כל תא יש לו תוכנית מסוימת. תא בעור יבטא גנים מסוימים, תא דם יבטא תאים מסוימים, תא במוח פה יבטא תאים כאלה, פה תאים אחרים. אוקיי? מתוך ה-20,000 תאים מתבטאים כמה אלפים בטח בכל תא. וחלק מושקטים וכו'. עכשיו, יש גנים שמופיעים בכל הגוף שלך, ולא מתבטאים בשום מקום, נניח ברטינה, אוקיי, רודופסין, רק ברטינה, רק בתאים של הרטינה. כל שאר הגוף אין לך רצפטורים לאור בקילון, אוקיי, או באור, או עליהם. גם פה חיפשו את הרצפטור, אבל גילו לא 1, 2, 3, גילו 1,000. שזו הייתה תגלית שהייתה מאוד דרמטית ביחס לחשיבות של החוש הזה.
לקח לה 8 שנים, זה גם...
מה?
כי לקח לה 8 שנים, זה...
לה לקח 8 שנים?
זה התמדה מטורפת.
אבל לי במעמד שלכם, שתבין. הרבה יותר זה נובל, זה החלטות שאתם צריכים לקחת. מה תגלו, ואם תגלו, אז זהו. אתם עכשיו רק צריכים להחליט מה אתם רוצים לחקור. אני רוצה טישו.
אני לא... כן, אני בינתיים כלום. תראו, בואו נעשה, אני לא יודעת מה זה מגנן, אני אגיד לך את האמת? לא.
כדאי לתת לי heads up כדי שאני אכין את זה.
אני אומרת, תשלח לך איזה זמן המדובר, ואז נקבע מועד קבוע, ואז שיהיה לי כמה דקות.
מה זה טוב מאוד?
אני שבאמת לא מתה על הקונספט הזה של ההפסקות, אבל...
כן, רציתי לשאול מה דעתכם על
אני הייתי
עדיף
מנועה
ההרצאות?
אני הייתי מנועה לעשות. נראה לי לא מבחינת...
יש לך כסף של הקבוצות הרצון.
אהלן.
להשתמש בלפטופ, כל מיני לעשות להתחבר מהעמדה.
מה זאת אומרת? אתה יכול מה שאתה רוצה, כל עוד אני יכול ללמד זה בסדר.
כן, זה תראה, פשוט חשבתי אתה משתמש במושל.
לא, אני משתמש אוקיי, יש לנו עוד כמה דקות, נכון? 3 דקות אתם.
אני יכול לשאול עוד שאלה, לא קשורה לשיעור? צריך לעשות את המבחן גמר שלי מתישהו.
איזה מבחן? אתה עובד בדוקטורט? אוקיי.
למתי פחות או יותר לחכות לחזור אלינו משהו?
הבעיה היחידה מתחילה כשאנחנו מתקרבים לסוף השנה. כי הם סוגרים שנה ב-31 בדצמבר, אל תגיד לי את העבודה ב-20 בדצמבר.
לא, את המאסטר כאילו, את התזה אני כבר אחר כותב, זה לא האישו, אבל המבחן.
זה כן האישו, אני לא נותן לך מבחן, עד שאני לא מקבל אישור מאלונה שעשית את כל הקורסים, ושאני יודע שיש לך מאסטר כתוב. אם אודי אומר לי, תשמע, אנחנו שבוע הבא מגישים, אז אני יכול להתחיל להרגיע. לוקח לי משהו כמו, ברגע שאתה מגיש את התזה, לפחות חודש לבדוק את זה, לפחות למצוא מישהו שיבדוק וייתן תחתים אחרים, ולארגן פגישה במבחן, זה גם כן משהו כמו חודש. ברגע שאתה בא ואומר לי, אני מוכן,
הצלחתי לסיים בשישי, אז בשמיני פחות או יותר, אבל מה הדדליין האחרון? מצד דצמבר, אתה אומר?
מבחינתי לי אין דבר, תעשה מתי שאתה רוצה. אני רק אומר לך שאם אתה לא מגיע לדצמבר, צריך לשלם שכר לימוד, או מה שנה הבאה. אוקיי.
I'm gonna go out of this lesson and go back to it. You wanna start? Remember this is mock. Now you guys now know my secret place. My locker, like open locker.
יכול להיות שנניח אני עשיתי את זה קצת לא טוב, כי באתי ופשוט דיברתי במקום, אולי אני צריכה
נהל דיון.
שכל אחד יכין שאלה
כל אחד אפשר לעשות לעשות. אנחנו נעשה את זה עוד פעם, כי אני לא הייתי נוכח, אני רוצה לראות את ה... אנחנו נעשה לך ניסויים. אתם מרוויחים את זה מבחינת מה?
כל עוד לא מטרידים אותנו בסוף.
לא עושים עליכם היסטולוגיה, אתה אומר. אחרת מי יעשה היסטולוגיה על Okay, so this just basically shows you that when you knock out the ion channel, the scat ion channel, you can't get any responses. So this is the wild type, and responses from the nerve of the axons of the olfactory system. These are different odors, and they elicit a very strong response. In knockout, it's flat completely. So these are anosmic animals. Okay, so let's talk just a little bit about these olfactory receptors. And like I said, these are part of the multi-family of the G protein coupled receptor, the 7 transmembrane domain, meaning that they cross the membrane 7 times and then form a complex structure. And as I told you, the amazing discovery was that this is a family of 1,000 genes out of 2,000 genes, 20,000 genes. Okay, so this is a huge family as compared to what we think the body has to do. Okay, and they have some homology. This just shows where are the regions that are highly variable and where are the regions that are conserved. Most of the conserved regions are within the membrane, in the outer side of the receptors, like the left and right sides here, and the more variable parts, most probably the ones that have to interact with the others, the complex others in the environment, are in the middle and outside the membrane. Okay? If you look into the genome, these proteins, again 1,000 proteins, are not specifically stuck in one chromosome or another. Okay? They are all over the chromosomes. So it gives you a very challenging task now, and you'll see why a little bit later in a few slides. But if you want to map where are these receptors, they're all over the genome. All of the chromosomes, there are even a few on the X chromosome, and none on the Y, but nevertheless almost all over the genome, with some kind of concentration on chromosome number 11 for unknown reasons. This is the relative amount of receptors out of the 1,000. Okay, so these are the specific loci on the chromosome and the number of genes in that area.
So what smells can females have and or
No, both. I have an X and Y. You have
no, I
two
know,
X.
but like if the amount doesn't like I
No,
don't
we
know
have the
what-
same number of receptors. Females and males have the same number of receptors. Okay. Great. So they're all over the place in the genome. They're all 7 transmembrane domain. We talked about that. And one interesting thing about this is that still today in 2016, we say that these receptors are orphan. And when we say orphan, it means that we don't really know what is the ligand of these receptors. It's odors, but we could not really identify which odors are activating which receptors. Okay, so Gal's idea to do a quick mapping came out very difficult to do in fact. And if you look at all other GPCRs in the genome, okay, if you look at the human, for example, let's look at the mouse, okay? There are 1,000 olfactory receptors, which are the G-protein coupled receptors. This is a Nobel Prize winning discovery of Linda Buck. There are many other G-protein coupled receptors, okay? Something like 400 in the genome of the mouse. Out of the 400, we know the ligand of about 200, 250 in the body, okay? All kinds of molecules in the body that we know that are activating, again, in physiology, if you took the course, adrenaline, noradrenaline, all kinds of neurotransmitters, etc., that are activating G-protein coupled receptors, and we know what is the ligand, what's the molecule activating it, downstream, etc. In the world of other receptors, we know very few ligands, and all the rest are orphans. They have no parent that activates them. We don't know it.
Why is this so difficult? I mean,
What?
I can think of few ways to do it. Why is this?
How would you do it?
Overexpress it.
Fantastic. Okay, so Gal is already in the industry of overexpression. What normally a scientist would do is use heterologous systems. Okay, so you take cells that express well, like HEK cells. You heard of HEK cells? Human embryonic kidney cells are very good to work in the laboratory. So you can plate them on many plates and now express different receptors. HEK. What? Yeah, there are other types of cells, okay, that you can use, of course. The problem is that when you express the proteins, they are not getting shuttled they're getting stuck in the cytosol. And they never get shuttled to the membrane. So you can express them. What?
Can you explain what does it mean?
So normally this is a protein which sits on the membrane of the cell, right? So this is the cell, okay, let's say the cell body here. There's some dendrites, whatever. Actually, the dendrites are important for us. So let's say this is the cell body, this is the axon, and now there are, dendrites of these cells. Okay? And the receptors, they sit on the membrane. These olfactory receptors, they sit on the membrane to detect the odors, right? They have to interact with the outside world, and these are 7 transmembrane domain proteins. I just tell you about these they sit in the membrane. The issue is that you can infect these cells and express this DNA, okay, this gene, and the protein will be expressed, but it will not go to the membrane. Apparently, you need specific machinery that will shuttle the protein into the membrane. And this you must have if you want to have these heterologous systems to do some kind of screening.
We know, like, you can add a sequence afterwards. That is a sequence that.
Shuttle in sequence.
Yeah.
So this did not work. Your simple solution did not work, but in 2004, 10 years ago, there's a special family of proteins that their role is to provide escorts to the olfactory receptors to the membrane. These are called the RTP family of proteins, and once they were found, it opened the world to do screening for these receptors. Okay? Now, still not super easy because you now need, let's say, okay, so now you need to express the olfactory receptor. You need to put these RTP molecules together. Only then it will be in the receptor, in the membrane, sorry. And now you have to play odors, and you need some output from the cell. Either you stick an electron. What normally people do is that they put a reporter, a fluorescent reporter. So you can put it on plate and start to. Okay, so people only started doing that in 2004. And I think maybe 3 or 4 labs in the world wide do that. Okay? Still, you live in an era, you can choose whatever you want to do. If you think the most important question in neuroscience is to de-orphan the receptors, I can recommend the lab that you can go to. Okay? Okay, so we're still at the point where we don't really know. Most of the community works with very few receptors that have been de-orphaned, so we know what the molecules that activate them, but we don't have a full-blown image of the real world of activating the receptors, although Gal claimed initially that this will help us to understand odor space better. Okay, so if you look at this family, there are 3 things that pop up which are interesting. One, there is dramatic change in the gene repertoire between different animals. There's large number of pseudogenes, we'll talk about all these 3, and large variation within species, which is also interesting.